Assalamu alaikum students. Welcome to microbiology lecture series. In today's lecture we will learn about internal organelles of a bacterial cell such as mesosomes, glycocalyx which consist of capsule and slime layer, cytoplasm, ribosomes and a bacterial nucleus. Moreover, some important questions related to this topic are also included in the end of this lecture. So let's first discuss about the mesosomes. What are the mesosomes? Mesosomes are also called as chondroids, are the intracellular membranous infoldings of the cell membrane. These are found in association with nuclear area or near the site of cell division. Mesosomes are absent in eukaryotes. These are formed due to vascularization of lipid bilayer, and the mesosomes are the special components of cell membrane, but the proteins differ from the cell membrane. Structure of a mesosome. As mesosome is the infolding or folded invagination of the plasma membrane. So if you look at the bacterial cell, these are the infoldings of the plasma membrane, means mesosomes. Further, here is the enlarged or separate structure of a mesosome. This is the cell wall of a bacterial cell, and this is the plasma membrane layer. Now this whole enfolding of the plasma membrane is called as mesosome. This is the mesosome sac, and these are the tubules. More importantly, the exact structure of a mesosome is unclear, but they may appear as lamellar, tubular, and in vesicular form. Mesosomes initially were known as internal bodies. A scientist, Greenwald, in 1975 named these infoldings as mesosomes. These may take part in respiration. However, these are not analogous to mitochondria because mesosomes lack the outer membrane. These are thought to be involved in cell division, as during the binary fission a cross wall is formed and resulting in formation of two cells. Mesosomes increase the surface area of plasma membrane, that increases the absorption of nutrients. Mesosomes also secrete certain enzymes such as penicillinase, which destroys penicillin. So now we will discuss about the glycocalyx, which is divided into two types such as slime layer and capsule. So what is glycocalyx? How can we define the term glycocalyx? Glycocalyx is a viscous, thick gelatinous material accumulates on the external surface of the cell wall and surrounds the cell. Glycocalyx is composed of polysaccharides or polypeptide depending upon the type of bacterial species. It is divided into two types such as slime layer and capsule. So let's first discuss about the slime layer. What is a slime layer? Slime layer is formed if the glycocalyx is disorganized without any shape and it is attached loosely to the bacterial cell wall. It is a water soluble, that's why bacteria show viscous colonies on media. So if we look at the figure, this is a bacterial cell and this is a slime layer. The disorganized without any proper shape surrounds the bacterial cell. Now capsule. What is a capsule? It is a type of glycocalyx and formed if the glycocalyx is well organized in a proper or defined structure. So if we look at the figure, this is a bacterial cell and this outer defined organized layer is a capsule. Capsule is further divided into two types such as homopolysaccharide capsules and heteropolysaccharide capsules. Homopolysaccharide capsules are usually made up of a single kind of sugar such as glucose sugar, whereas heteropolysaccharide capsules contain more than one kind of sugars such as galactose, glucose and ramenose. Functions of a capsule. Capsule helps in adherence to various surfaces of plants, roots, human teeth and many more other surfaces. It helps bacterial cell against temporary drying by binding water molecules. Capsule serves as a reservoir of the stored foods. It prevents bacteria from bacteriophages. Capsule protects the pathogenic bacteria from being engulfed by WBCs such as phagocytosis process. More importantly, capsule is in antigenic nature. Now cytoplasm. What is a bacterial cytoplasm? Cytoplasm is a gel-like internal matrix of the cell inside the cell membrane. So if you can see the bacterial cell structure, here is the internal matrix which is called as cytoplasm. It consists of water, proteins, carbohydrates, inorganic ions and other low molecular weight components. Cytoplasm consists of DNA molecules, ribosomes and other inclusions. It is a protoplasm where the functions of cell growth, metabolism and replication of a bacterial cell are carried out. Now ribosomes. Ribosomes are the protein synthesizing organs of a cell. It consists of RNA and proteins. Ribosomes consist of 30% of the total ribosome of a bacterial cell. A prokaryotic cell may contain about 10,000 ribosomes. 70s ribosomes in prokaryotes, while in eukaryotes it's 80s. Here S refers to the Svedberg unit, which mediates the relative ratio of sedimentation during the ultracentrifugation. Mainly, the sedimentation rate depends on size, shape and weight of the particles. If we differentiate between eukaryotic and prokaryotic ribosomes, so an important point to be noted here is that the eukaryotic ribosomes are found attached with the cell membrane or plasma membrane, but the prokaryotic ribosomes are found freely in cytoplasm. Moreover, another important property of ribosomes is that ribosomes are not specific in nature, means ribosomes of one species can be used for the protein synthesis in other species. Bacterial nucleus. What is a bacterial nucleus? Bacterial chromosome or nucleus is called as nucleoid or chromatin bodies due to absence of nuclear membrane. A true nucleus is absent in bacterial cell. A bacterial nucleus or nucleoid is a single long circular double-stranded DNA molecule without histone proteins. Appear as a clear area with a mass of fibers, and it is a gene bank and stores hundreds of genes which control all the functions and properties of cell. Bacterial nucleus plays a role in inheritance properties of the cell from one generation to another. Now questions. What is difference between capsule and slime layer? Who named the enfoldings of the cell membrane as mesosomes? Which enzyme destroys penicillin antibiotic? Which bacterial structure serves as a reservoir of stored foods? Why bacteria show viscous colonies on media? What is difference between homopolysaccharide and heteropolysaccharide capsules? Why bacterial chromosome is called as nucleoid or chromatin bodies? Why are the ribosomes called as protein synthesizing organs of a cell? Ribosomes of one species can be used for the protein synthesis in other species. How it is possible? What do you mean by protoplasm? What is difference between DNA and RNA?